Charles, would you be able to tell us a little bit about your talk?
Certainly. So I endeavoured to give a broad overview of recent advances in what we understand about the genomic landscape of acute lymphoblastic leukemia in both children and adults. Some of the key messages were that we've now studied several thousand cases with some level of genome sequencing or transcriptome sequencing, and that's shown that firstly there are a number of new subtypes that were previously unrecognised, as well as striking variation in the frequency of subtypes according to age. Particularly some of the good risk subtypes in children are no longer present in adults, and some of the newer subtypes, particularly kinase-driven leukemia, are predominating in older adolescents and older adults. Another point was the striking heterogeneity in some of these new subtypes. Typically, each subtype of iwAL we still consider to have a distinctive gene expression profile. But in contrast to previously described subtypes, where often there was only one translocation associated with a subtype, we now often have one fusion partner rearranged to many different fusion partners in a given subtype, or indeed, such as in PH-like leukemia, a broad diversity of different fusion partners that converge on a given signalling pathway or pathways. The presentation also made the point that we continually need to optimise our classification. We've shown that in the context of immature leukemia, there are several newer subtypes, similar to ETP-ALL, that have their own defining genomic lesions, and these really should be considered as to be founding events in new subtypes of acute leukemia. The presentation touched on T-ALL and showed that from large sequencing studies there are a high frequency of some of the common mutations that were previously recognised, as well as a fairly high frequency of signalling pathway mutations that are, on one hand, strongly associated with individual subtypes of T-ALL, and secondly, quite frequent and often clonal, suggesting there may be good targets for therapy. They include the PI3K, JAK-STAT and RAS signalling pathways. The presentation touched on recent insights into understanding the role of germline genetic variation, showing that there are a growing number of genes that are found in familial leukemia, which is rare, but instructive as they often identify genes with a key role in leukemogenesis, as well as leading to studies showing that these genes are often found to have variations in sporadic leukemia in the germline. So there's more to germline predisposition than just looking at familial leukemia. And finally, the presentation looked at issues of evolution and genetic heterogeneity and how it influences treatment response and progression. It overviewed the landscape of mutations that are enriched at disease progression and relapse, and also touched on a couple of singular examples where individual genes and their mutations are associated with resistance to individual agents, such as CREB binding protein gene mutations in glucocorticoids and NT5C2 and response to thiopurines. And this has two implications: that we should be trying to detect some of these mutations early on in therapy and predict agents to which the patients might become resistant, but also they suggest that there are potentially new ways to circumvent resistance or to restore sensitivity in an additive or synthetic lethal approach with agents to which the leukemic cells are resistant. So I think broadly we had a very interesting session and a good coverage of a number of highly topical areas that are interesting both in terms of basic biology but also right at the cutting edge of how we think about integrating genomic profiling into clinical management. Perhaps I can come back to you, Kathryn. Can you comment on the challenges of testing for PH-like leukemia and how that can be done?
Absolutely. So that's, I think we've done a good job of laying out the genomic landscape. As you allude to, the challenge is how do we implement that in a clinical setting to identify these patients up front. And so we can take several different approaches. Obviously this is complex because of the many different alterations just within this PH-like subset. But I think with patients that are treated at a single centre, we can take approaches such as a candidate approach. We know the different alterations that are present, and we can implement assays to detect those alterations. This can be done at a small kind of single centre level platform. The caveats of this are that they only identify the alterations that we know, and so it won't identify the other alterations that we haven't discovered yet. And with each discovery exercise, we know that we identify new alterations. So, but that's, I think, a relatively simple approach that we can take at single centres, be it FISH assays or RT-PCR-based Sanger sequencing approaches. Places like the Children's Oncology Group, which are testing these patients on a national level, so they test thousands of patients, are taking a screened or a tiered approach where they screen up front for the gene expression profile of PH like ALL based on a very simple TACMan array card that has 15 genes that are highly expressed in PH like ALL. There's been a lot of optimization to reproducibly identify these patients. They screen those patients and then they perform comprehensive testing on those patients downstream, just purely because they can't perform the comprehensive testing on all patients at the moment. So that's kind of more of a tiered approach. And then I think really the way of the future is doing a more comprehensive, unbiased sequencing. So, for example, employing whole genome or whole transcriptome sequencing to really identify the full spectrum of alterations that may be present in patients. And centres like St Jude Children's Research Hospital is implementing this approach. So I think it depends on how many patients you have, whether you're a single centre or managing multiple different centres, and what capacity you have for genomics. There are different approaches available. I do think in the future we will be going to more of the unbiased genomic sequencing, and that's something that we're working towards.
It's a good example of the challenges of diagnostic testing, and another consideration, of course, is what agent you're seeking to allocate patients to. Anjali, what about managing ETP in the current era, given that it's a relatively uncommon entity? How do you envisage this should be coordinated within a centre or between centres and across cooperative groups? What are the approaches that you're considering or taking?
That's a great question. So I think right now, until we have more data available, one question that came up at the meeting was, so should you transplant these patients in first remission? I think for the data we have right now, and from the studies that have been done thus far, it looks like using an MRD-based approach to make that decision is probably appropriate. I think the harder question is, at what intervals do you need to check MRD after the initial MRD if they're negative, and what is the relapse rate after that? I think those are questions that need to be answered. I think that because ETP is such a rare subset, it's really going to be important for larger studies on the cooperative group level or national level, really to answer a lot of these important questions. For example, one of the questions that came up was about the addition of nelarabine in adult ALL, and I think, again, that's going to be very important. Right now, all we have are phase 2 studies, and because of the rarity of this disease, it's really going to take cooperation both on a national and international level, I think, to answer a lot of these important questions.
And of course, ETP-ALL has its own diagnostic challenges because it isn't a genetic diagnosis. It's currently diagnosed by flow cytometry, and there's this related but not identical entity called near ETP, in which one of the markers that we use to classify ETP is somewhat different, and that's CD5, which is now present. How are you considering those cases, which are not that infrequent compared to ETP-ALL? Do you put them in the same group, or do you treat them as conventional T-ALL?
That's a good question. So I think for both, right now, you have to really take a very careful MRD-based approach and then make your decisions about transplant based on that. So in general, we think about those as maybe not as high risk, but again, I think if a patient has a very high level of MRD after induction, those patients are probably as high risk as those with ETP-ALL.
Okay.
And if I could ask a question of you, Charles. So you've identified some new subtypes based on rearrangements such as MEF2D and ZNF384. How will they be implemented in the clinic? Will they drive risk stratification? How do you see this information being implemented? So if we identify a case with a kinase alteration that's kind of obvious, they may be candidates for TKI therapy, but how do you envision if a clinician identifies some of these newer rearrangements, how does that change therapy or not, or risk stratification?
It's a very good question. I think one that we're all grappling with because we're continuing to identify these cases. Not all centres can identify these fusions, which are often cryptic on cytogenetics, and they vary in terms of their fusion partner. And we don't have large numbers of cases across many protocols. So I think it's fair to say, looking at the landscape of clinical trials that are now opening or are planned to open in both children and adults, we're still taking a fairly focused approach in that many groups are using pH like iwAL as an initial risk stratification and therapeutic targeting decision point. But many of the other genomic features are not yet used for initial classification or changing of the initial intensity of therapy. But that may change over time. There are some examples where some of these entities that you've mentioned have their own particular potential therapeutic vulnerability, and it may be over time that if these cases respond poorly, then additional novel targeting approaches may be brought to bear on those cases. Or indeed, as we accrue more evidence as to the association between a new subtype and outcome, because some of these are considered to be poor outcome from retrospective studies, one might consider intervening at an earlier time point with a newer approach, whether it's a targeted agent or one of the newer immunotherapeutic approaches. So I think the simple answer to this is watch this space, but they will likely have at some level an impact on the way that we manage and initially risk stratify our patients. Well, thank you. We've had an excellent session. This has really been outstanding in giving a broad overview of some of the most important advances in the molecular genetics of ALL, and with a very strong emphasis on the diagnostic challenges and potential for new therapeutic approaches. And so as we move forward, we look ahead to seeing these approaches implemented in the clinic.